Hello students, now we will discuss about a new topic, that is point mutations. What are point mutations? Point mutations are those that are caused by change in any one point of the nitrogen base in a gene sequence or in a DNA. Point mutations are of two types, that is base pair substitution type of mutations and base pair addition or deletion type of mutations. Here, base pair substitution type of mutations are also called as indel mutations. This is another term given further. This is of two types again. One is transition type of mutations. Another is transversion type of mutations. Now, this transition type of mutations means the substitution of one purine by other purine or one pyrimidine by other pyrimidine. But here in case of transversion, one purine which is normally present, it can be substituted by other pyrimidine. So here I am going to write the meaning of the transition mutation means the mutation caused by substitution of a purine by other purine or a pyrimidine by other pyrimidine. Normally when we are discussing like this, we will just write vice versa. Okay, pyrimidine by other pyrimidine. In case of DNA, we have two types of purines. One is adenine and guanine. In DNA, there are two types of pyrimidines. Those are thymine and cytosine. These are the two types of pyrimidines and these are the two types of purines. But in case of RNA, this thymine is replaced by uracil. Now, here in this case of transition type of mutations, examples discussed are beta thalassemia. In the previous videos, I told what is meant by thalassemia. Thalassemia, one type, is already discussed in chromosomal type of mutations, that is deletion type of mutations, which is nothing but severe anemia that is caused by non-formation of oxygen-carrying hemoglobin. When hemoglobin is less, then oxygen is not evenly or properly supported all over the body, then the person will be prone to anemia. When this hemoglobin is lacking, and then what is linked between hemoglobin, oxygen and anemia? Hemoglobin molecule is having ferrous ions, ferrous molecules. When ferrous is not there, then a person will be obviously prone to anemia condition. Another example is phenylketonuria. Now, these two types of disorders or the type of transition mutations are their genetical codon sequence is different, but the type of amino acid is the same. These are also called as silent mutations. These are also again called as synonymous mutations. Now, in this case, if you see examples for silent mutations, we can write GAG is one codon sequence, guanine, adenine, guanine, that is coding for glutamic amino acid. Now, here this purine is replaced by other purine during the mutation and it is giving out another codon sequence, that is guanine, adenine, adenine. It also codes for glutamic acid only. Such type of mutations are also given a term like silent mutations. Next, another what is meant by transversion type of mutation. The mutation caused by substitution of one purine by pyrimidine and vice versa. Means a pyrimidine by a purine. This means a pyrimidine by purine or purine by pyrimidine. That is in reverse. Here examples we can take sickle cell anemia. Sickle cell anemia can be taken as best example because in case of sickle cell anemia, at 6th codon sequence of beta chain of hemoglobin, at 6th position, we have the sequence like GAG, which is coding for glutamic acid. But there, instead of this GAG, adenine means it is purine. Now it is getting replaced by a pyrimidine, that is uracil. So if this adenine is being replaced by uracil, then it codes for other amino acid, that is valine. So this can be considered as transversion type of mutations. So under transition type, we have seen two examples or two disorders, that is beta thalassemia and phenylketonuria. Under transversion type of mutations, we can see the examples like sickle cell anemia. Next, about another type of mutations, that are base pair addition or deletion type of mutations. Next is base pair addition or deletion type of mutations. Here, because of addition of one extra nitrogen base pair to the existing triplet codon. We know that one triplet codon codes for one amino acid, whatever it may be. Because of that, that particular triplet codon is not divided by 3 number. If either by deletion or addition, the same thing happens. Here what happens, for suppose take out one extra type of amino acid like UUU, means uracil, which is coding for phenylalanine amino acid. Next, take out another amino acid like, codon sequence that is ACC. Because we are telling these are codons, and this will be coding for amino acid that is arginine. Now, for suppose think that one extra nitrogen base is added here like this, then also it will not be divided by 3. If one nitrogen base is deleted, then also it is not divided by 3. And so that what happens? When it is not divided by 3, that particular triplet codon, so and so amino acids are not synthesized either in this case or when addition type of mutations happen. Such type of base pair addition or deletion mutations will lead to total change in the frame of triplet codons, which will cause changes during translation. So we can tell base pair substitution mutations either by addition or deletion of one nitrogen base, the frame of the total translation process will be changed. The frame of total translation process, or we can say simply translation process. Translation process is disturbed. And so what happens? The proteins are not formed up to the mark. Proteins are not correctly framed. This not correctly framed, what does it mean? It means that total protein structure will be lost. It means the loss of original protein structure will be caused. When a protein has lost its original protein structure, obviously its functions also will be disturbed. So this can be considered as one best example for frame shift mutations. Means base pair addition or deletion means nothing but frame shift mutations. We can also give one more example by addition or deletion, which is given in your NCERT textbook, is also one of the best example. But here I will write one more example. If the first amino acid is coding for threonine, we can take amino acid coding, first codon. It will be AUG, which codes for methionine. Next, it is attached with the help of a peptide bond to another codon sequence. Here not peptide bond. Here amino acid, between amino acids is peptide bond. Here between the codons, there is next subsequent codon. Let's take one example like ACA, which codes for threonine. Another codon sequence, that is GUG, which codes for valine. Here will be peptide bond. Another codon sequence we can take example like what shall we take? UUU. Okay. UUC, which codes for phenylalanine. For suppose if there is an extra addition of any type of nitrogen base at one point of the codon, then it is also not divided by 3. And so what happens? During the translation process, if any mutation was observed by the DNA, that particular codon will be eliminated from the newly formed DNA strand if it has to be cured. If it is not eliminated, then the mutation will be continued as it is. If any one nitrogen base was deleted or removed, then also the total framework is changed. Okay, so this methionine initiating amino acid by the codon is not synthesized. Then also there will be complete disturbance in the total protein structure. So this frame shift mutation is very easy to remember also. One best example was also given in your NCERT textbook. You can go through that. We will see some other type of mutations. One is missense mutations. What is meant by missense mutation, which is also termed as antisense mutations? These are some of the terms that are used while asking the previous MCQs. Antisense mutations, which is also called as non-synonymous mutations. Now, if a new codon is encoding different amino acid during the transition mutation, due to the transition mutation. Now, what does this 3 words means? The new codon that is changed because of the mutations codes for different amino acid when we compare to the original sequence. Amino acid due to transition mutation. What is transition mutation? Replacement of a purine by purine or replacement of pyrimidine by other pyrimidine. So here examples we can take. For suppose UUA sequence of the codon is coding for leucine amino acid. Then if there is any change in the uracil, means one pyrimidine, if it is replaced by another pyrimidine, that is cytosine, then it codes for serine type of amino acid. So this is called as missense mutation. Another example is nonsense mutation, in which the new codon that is formed due to mutation is a stop codon. All these type of new codons that I am mentioning here are due to base pair addition or deletions. Now, here examples we can take as CAA type of codon sequence, means cytosine, adenine, adenine. It is coding for one amino acid, that is glycine. Then, if there is change in any one, either purine or pyrimidine, here according to the sequence I am changing the pyrimidine, that is cytosine, it is replaced by uracil. Uracil is also a pyrimidine. Again, we can consider this is a type of transition mutation again. So it is coding for UAA codon sequence, which is considered as stop codon. Now, there is a name for this UAA type, that is occur. Now, there are some other types of stop codons. So stop codons in genetic code are 3 in number. One is UAA, another is UAG, which is also called as AMBER. This AMBER spelling is AMBER. Now, there is another type of stop codon, that is UGA. This is also pronounced as AMBER, but the spelling is different, which we also called as OPAL. Okay, so these are the three types of stop codons, but we are discussing about only one type of stop codon that is framed out because of the nonsense mutations.